Yeah, welcome to another session of Bioinformatics. I am Dr. Y.V. Rajesh, Assistant Professor, Department of Biotechnology. In today's session, we'll learn about the forces stabilizing the protein structure, which is a part of structural bioinformatics. So, the forces which stabilize the protein structure, there are basically four main types of forces are there. Four main types of non-covalent forces here. So, hydrogen bond, ionic bond, van der Waals interactions and hydrophobic interactions. So, in this graph you can see the energies of different types of bonds here. So, different types of bonds in kilocalorie per mole. So, you have thermal energy, van der Waals energy, electrostatic energies, hydrogen bonding energies. So, all these four energies are called as non-covalent interactions here. And beyond this, the covalent bonds are there. So, maybe for single bond and for double bond, and hydrolysis of ATP for phosphoanhydride bond. So, this is the energy given. So, for protein structures, H bonds, ionic bonds, van der Waals and hydrophobic bonds are there nothing but combinations of van der Waals interactions, and also affinity of some of the proteins, affinity of some of the amino acid residues to avoid water here. The bond energies range from 1 to 5 kilocalorie per mole here. So, in primary structure, proteins basically they are made up of peptide bonds and disulfide bonds. Only covalent bonds play a role for the protein structure, which is a linear arrangement of amino acid residues. And for secondary structures where we have alpha helices, beta sheets and turns, so hydrogen bonds, peptide bonds and disulfide bonds. So, additionally hydrogen bonding will be added up to the primary structure. And for tertiary structure, along with hydrogen bonds, hydrophobic interactions will also and electrostatic or ionic interactions, van der Waals interactions will add up. Additionally, hydrophobic, ionic and van der Waals interactions will add up to form the three-dimensional structure. And for quaternary, so more of the same bonds will be there, but additional hydrophobic interactions will be added here. So, we will focus on each force step by step here. The first one is electrostatic interactions. The electrostatic interactions basically they function between the positively charged and negatively charged atoms when they are separated by 15 angstroms. 15 angstroms, the positive charge will be attracted by negative charge here. So, in amino acids we have COOH which is negative and NH3 plus which is a positive here. So, they make a attract with each other if they are not bonded together here. Basically in the middle actually they are bonded together by covalent bond, but some of the amino acids such as lysine, arginine, aspartic acid and glutamic acid, in their side chains they can have these negative and positive charges. Lysine and arginine have NH3 groups. Lysine will have epsilon amino group and arginine has guanidine group. So, both are positively charged here. And aspartic acid and glutamic acid has beta and gamma carbonyl carboxyl groups which are negatively charged here. So, these things when they are present, when a positive charge and when a negative charge, they are located by in between 15 angstrom unit distance here. They can form interchain electrostatic interaction and intrachain electrostatic interactions. So, more than one electrostatic interaction, we can call them as a salt bridges here or ionic bonds here. So, basically these are ionic bonds and more than one interaction will become the salt bridge between two different chains of a protein here. So the next one is hydrogen bonding here. The hydrogen bonding basically it operates less than 3 angstroms. So less than 3 angstroms only they can operate here. So there is a hydrogen bonding is nothing but the interaction between a partial charged atom to the H, the hydrogen which is attached to the nitrogen here. So when you have oxygen and the nitrogen, so the nitrogen which have the partial charge here, they can form the hydrogen bonding here. So basically it is between OH, O and H, S and H, where H is connected to nitrogen here. So and which is operated between less than 3 angstroms. So we already learnt about hydrogen bonds in school days, and more detail also we have seen in our +1 and +2 here. So basically the hydrogen bonding operates at a lower distance range here, and because of hydrogen bonding the alpha helices are formed. So alpha helices are basically because of intra-chain hydrogen bonds between 1 to 4 to 2 to 5 3 to 6 here. So the arrangement of side chains is shown with green color, which can form the hydrogen bonds here. And similarly beta sheets also, the hydrogen bonds between parallel chains and anti-parallel chains here. And only proline, the amino acid proline, which cannot fit into alpha helices or beta sheets, it can form into helix breaker here, and helix breaker because it does not have any hydrogen bond donor here. And next interactions, those are called as van der Waals forces. Those van der Waals forces contribute to the overall protein stability. They will contribute to the overall protein stability. Basically any two atoms which are less than 2 angstroms and not more close than 1 angstrom distance, they will be attracted. Forces will be attractive forces and repulsive forces based upon the distance between them. So when several set of atoms they come close together, they can show attractions or repulsions here. So at bigger level van der Waals forces will become hydrophobic interactions here. So these are instantaneous forces, because when atoms will become transient dipoles, and the oscillating dipoles can result in attractive forces here. So here is a diagram of lizard sticking to the walls because of the van der Waals interactions. So compared to the hydrogen bonds or electrostatic bonds, van der Waals interactions are weaker, but however they play key role in protein structure formation here. So same way like electrostatic interactions, it can be intra-chain van der Waals interactions and inter-chain van der Waals interaction can be observed here. And next force which may not be directional, like hydrogen bonding or electrostatic interaction, may not be specific, but has most important feature in protein shape formation and structure formation, called as hydrophobic interactions here. Basically there are two types of amino acids are there: hydrophilic amino acids and hydrophobic amino acids. So the hydrophilic amino acids means which can form hydrogen bonding with the water in water solution, and they try to like water, try to have interaction with water, whereas hydrophobic amino acids, which they don't have the capability to form hydrogen bonding, so they try to avoid them here. So in this case the green colored ones are hydrophilic amino acids and red colored ones are hydrophobic amino acids. So they can form into this kind of shape, where hydrophobic will go to interior of the protein structure and hydrophilic amino acids lies on the surface of the proteins here. So hydrophobic amino acids are glycine, alanine, valine, leucine, isoleucine, methionine and proline and phenylalanine here. So the composition of hydrophobic and hydrophilic amino acids actually guides the shape of the proteins here. So there is sometimes in some proteins they have some specific coordinate covalent bonds are there. In zinc fingers, a zinc atom can stabilize some specific structures here. So similar zinc atom, manganese, magnesium and chloride ions in some specific proteins, they stabilize the protein structure by forming some sort of specific covalent coordinations between the atoms here. So these are the major forces stabilizing the protein here. So stabilizing the protein along with peptide bond and disulfide bond, and that is leading to the final structure of the protein. So based on the composition of the stabilized forces and amino acid compositions, so a protein can be linear, it can be the shape, it is a porous substance kind of shape, or it can be a question mark shape here. Very complex shapes can be seen here. A triangular or pyramid like kind of shapes can be observed in proteins here, because all these things are dictated by stabilizing forces present in the protein structure here. So thank you very much for listening to my class. Thank you.